to the cloud. It should be going now.
Okay, welcome to today's Polyploidy webinar. My name is Jeanette Witten, and I'm going to be introducing Michael McKibben, our first speaker for today. Michael is a PhD student at the University of Arizona, where he's working with our fearless leader, Mike Barker. He is going to talk to us today about the legacy of whole genome duplications in diploid plant genomes. This is a fascinating area, trying to sort out what the lasting effects are of those macro mutations that we're all so very fond of. I wanted to mention, if you didn't get a chance to read Michael's bio, that he has his undergraduate work at the University of Tampa involved studying sarcoglossans, a group of sea slugs. And so if you have a moment of attention at some point in the next hour, you could go over and do an image search on sarcoglossans and see something new, or maybe just remind yourself of a group of organisms that you haven't thought about until today. It is piss pouring rain here in Vancouver, and it has been and will be for, I don't know, the next 40 days at least. And so it's not hard to think about organisms that live in water right now. They all seem to be front of mind, just literally swimming down the street here. So with that, Michael, I'll hand it off to you. And as always, please mute yourself during the talk. You can put your questions and comments in the chat, and we'll pick up that thread in a little bit. So off to you, Michael.
All right, thank you very much for the introduction. To sort of repeat a little bit, today I will be discussing with you guys the legacy of genome duplications in diploidized plant species. And across plants, particularly angiosperms, ancient whole genome duplications are a surprisingly common event. For example, this is a phylogeny for the 1KP project, and every little red branch you see represents an ancient whole genome duplication on the phylogeny. A sort of fun way to think about whole genome duplication evolution, I always like the term polyploid hop. And so say we have a diploid lineage. Some members may experience a whole genome duplication and establish a polyploid branch. And there may be gene flow between these branches over time, perhaps even local speech extinction events. And for the majority of these branches, they may go extinct. However, the rare few will return to the diploid state through a process known as diploidization. And genes originating from this whole genome duplication can exist in the genome in a large shadow for millions of years. We call these genes Paleologs. And so say I have a hypothetical simple chromosome here. It may be duplicated, maybe even triplicated. And over time, some of these gene copies may be lost through the process of fractionation, leaving single retained, triple retained, or double retained Paleologs in the diploidized genome. Recent work by myself in my lab in Brassica rapa, a crop species, noted that candidate genes associated with domestication were significantly enriched with Paleologs. Similarly, genes that were differentially expressed between the many cultivars of Brassica rapa were also significantly enriched with Paleologs. And this got a series of questions in my brain surrounding this idea of how Paleologs might shape plant evolution post-diploidization. And that's what I'll be discussing with you guys today, with the first part of my talk being dedicated to how we actually identify them accurately within genomes of diploids, and part two moving on to sort of a first case use scenario of some of these tools for testing to see if this pattern of Paleolog enrichment in candidate domestication gene lists occurs outside of Brassica rapa. And then of course, wrapping it all up with some future directions. To begin the first part of my talk, I'd like to discuss some of the tools available to us today to identify Paleologs, including some very simple but relatively widely applicable tools, a much more precise but somewhat restrictive tool, a new tool I've developed called Frackify, which uses data from modeled whole genome duplications to train a machine learning algorithm to identify Paleologs. And then, of course, we'll be comparing these approaches and why you might want to use one over the other. To begin with sort of the simplest approach to identify Paleologs, we can use something known as a gene age distribution, Sometimes they're shorthand called KS plots, KS being the level of synonymous divergence between homologous gene pairs within a given genome. Peaks in these plots can be formed by whole genome duplications, and we can use complex mixture models in order to place bounds on these peaks. And it's generally assumed that genes within the bounds of these peaks are likely candidate paleologs. As you can imagine, this can lead to a lot of issues, particularly when there are multiple overlapping whole genome duplications, a pretty common occurrence in plants. And if we place bounds on these peaks, on the left-hand side, we'll get some false positives. On the right hand, we'll get some false negatives. And not all genes within the bounds of the peaks are necessarily paleologs. They might be the results of small-scale duplications. To help remedy this, we can use something called Syntony, which is defined as the conservation of gene order along blocks within chromosomes. We can represent these using something called a syntenic dot plot, where on the X and Y axes, we have each chromosome in our reference genome lined up against each other. And all these little dots represent a region of syntony. As you can imagine, whole genome duplications will produce quite a bit of syntony, which might be broken up through time. However, not all syntenic blocks are necessarily from our focal WGD. Once again, they can be from small scale duplications or perhaps multiple whole genome duplications within a given genome. Taking it a step further, we can also compare syntenic blocks to an outgroup. The simple as this being double conserved syntony, where say I have my syntenic block pair in my focal species. If both of these pairs also match to an outgroup, it's likely that they're a syntenic block from a given whole genome duplication. A more complex way of doing this is using something called quota-align, which uses a binary integer programming to try and sort out the origins of different syntenic blocks within a given genome. Between all these methods, it's pretty widely applicable. However, once again, they all suffer from issues of false positives and negatives. A much more sophisticated approach would be to use something called POINT, or the Polyploid Orthology Inference Tool. POINT is rather amazing as it uses syntenic information from multiple species and an outgroup in order to model complex duplication histories in a species. And so although POINT's original purpose is not for paleolog identification, for regions of the genome analyzed with POINT, we can make some pretty detailed inferences about paleologs, including their retention status, as well as which subgenome they might have originated from in the case of allopolyploids. The limitation of this is POINT is primarily run on syntenic blocks that are conserved between all of your species. So if I have the region of the genome where syntenic blocks have been lost in one of the species, I can't make inferences on paleologs within them. So this leaves me with a bit of conundrum for my future work on paleologs. I can use a very precise but somewhat restrictive tool or a very broad and approximate tool, and I was looking for something somewhere in between these two. For this, I decided to turn to machine learning with the goals of using only one genome and an outgroup, and using syntenic and KS information, as many tools already exist out there to identify this information within genomes, to not only identify if something's a paleolog or not, but similar to POINT, also give me information on the retention status of a given paleolog. But there's some major hurdles in accomplishing this task. With all machine learning, training data is a heavy limitation. In my case, simulating whole genome duplications and their entire evolutionary history in a diploidized plant genome can be very intractable if I want to cover all possible parameter space. And similarly, sort of what plagues all machine learning is any assumptions I make in my training data will necessarily be passed down to my later on predictions. So stepping back, let's think about what I'm actually simulating here. In this case, syntenic blocks. I have a hypothetical syntenic block here as an example, and all the collinear homologous genes within it are going to have a level of synonymous divergence. And I had some questions surrounding these syntenic blocks. How long are these blocks on average in diploid genomes? And what is the variation of KS values both within blocks and among blocks from different whole genome duplications? To start answering these questions, I used 27 genomes and ran MC-ScanX, a software for identifying syntenic blocks, on all of them and began answering my questions. To begin, I plotted the distribution of block sizes in the entire data set. So on the x-axis here, we have the length of synthetic blocks in genes, and on the y, we have their frequency within the entire data set. As you can see, the majority of this data is in 20 genes or less. However, there's a very long tail of much longer synthetic blocks. I was curious to see whether or not the age of the duplication might affect this distribution, as older duplications might have synthetic blocks broken up more through time. And to do so, I fit a log normal distribution to the distribution of synthetic blocks in each species, and then plot the standard deviation of these distributions against the mean Ks of the focal WD in each species. And as expected, we got a significant trend line with older duplications consisting of smaller synthetic blocks. Moving on to values of Ks, I started by observing the distribution of mean pairwise block Ks for each of the whole genome duplications. As an example of Brassica rapa, you can see that the youngest peak in it is relatively narrow, and as these peaks age, they get wider and wider. Once again, I plotted the standard deviation of synthetic block lengths against the peak mean Ks and got a significant trend. Finally, I did a similar analysis, however, this time looking at the standard deviation of Ks values within synthetic blocks, plotted against the mean block Ks, and fit a linear model, this time controlling for the length of a given synthetic block, and found another significant trend. Using all of this data, I wrote a basic Python script that simulated synthetic blocks you might expect from MC-ScanX results, which included modeling basic gene evolution and informing these models using these empirical distributions from real-world data to help avoid some of the assumptions I make along the way. And how well did these simulations do? On the left-hand side, we see actual MC-scan results from Brassica rapa, and on the right, we have a simulated Brassica rapa genome. And I might have gone a little crazy on the level of fractionation, but for the most part, it does a pretty good job approximating these distributions. Using this simulation framework, I then ran thousands of simulation across parameter space, including young whole genome duplications, old whole genome duplications, different numbers of whole genome duplications, and different levels of fractionation, and then use those to train a machine learning algorithm. As a reminder of what type of task I'm trying to accomplish, this is classification. I wanted to be able to tell not only the retention status of a given paleolog, but also whether or not the duplication is a hexaploidy versus a tetraploidy. After all, if I know in my genome I have an ancient duplication, but I'm getting a lot of inferred triple retained genes, something is clearly going wrong, and I need to go back and change how Frackify works. For this task, I used XGBoost, something known as gradient boosting, which essentially makes a lot of very weak or shallow decision trees and then slowly corrects for their weaknesses over time. To prevent overfitting, I used a Bayesian search across input parameter space to minimize something called the loss function, which you can think of as the trade-off between the accuracy of the model and the simplicity of it. Well, how did it do? Here we have something called a confusion matrix for all of the genes in the testing data set. On the x-axis, we have what the actual class of each gene in the training data set was, and then what Frackify predicted these given genes to be. So for example, for the non-Paleologs, 96% of them were accurately identified by Frackify, about 2% were misidentified as single retained genes, and so on. And as you can see across the multiple classes of Paleologs, Frackify maintained above a 96% accuracy. But one thing we need to keep in mind, this is across those thousands and thousands of data sets. It's possible that there are particular areas of parameter space where Frackify might struggle. For example, what if I have multiple WDs, something very common within plant genomes? They also might be closer or further apart, and older WGDs, as we reviewed before, might have broader peaks, all this leading to different amounts of overlap. How might Frackify do identifying paleologs in this overlapped region? To get at this, I split out all the simulations that had 3 whole genome duplications, sort of the most extreme example, and made another series of heat maps, however, this time split out by the retention status. This time on the y-axis, we have the mean KS of the focal WD I was trying to predict Paleologs for. And on the x-axis, we have how far apart the next closest whole genome duplications were. So say I took this little pixel down here, I was predicting Paleologs in a whole genome duplication at KS 1.8, and then about 0.1 KS away was then another WGD, and 0.1 KS away from that was a third WGD. And as you can see across the majority of parameter space, Frackify seems to maintain a high degree of accuracy, with accuracy breaking down as you get above KS around 1.0. One thing that's important to make note of in this figure is Frackify's misidentifying between retention statuses. However, even at this high degree of overlap and old duplications, Frackify very rarely predicts non-Paleologs as Paleologs. So this seems to be some relatively good news going forward. But as I mentioned earlier in the talk, this is all training data that I've generated through simulations. Does it actually mean anything on real data sets? So I wrote a pipeline in order to take annotated reference genomes in an outgroup. You can make inferences using either Goji SynMap PL or MC ScanX, and use those synthetic information to be reformatted in a way that Frackify could understand, and finally make inferences using Frackify. You can find details on everything I've reviewed in this talk so far in the Frackify pipeline on our preprint on bioRxiv, also available at this QR code here. And for real data sets to compare, I used a paleohexaploidy and a paleotetraploidy, and I used 6 species which had already had point run on it. As a review, point was the most sophisticated current method out there for identifying Paleologs, and I made additional Frackify inferences for these different species. I've picked 2 exemplar species out of the 6 to show you right now. On the left, we have a triplication in Brassica rapa. And on the x-axis, we now have points classifications, so non-Paleologs, single, double, and triple. And on the y, we have Frackify's classifications. I've done similarly with the duplication. And as you can see, for the Paleologs themselves, point in Frackify maintains around a 80+% agreement. But wait, what is going on with these non-Paleologs? Frackify seems to be identifying many more Paleologs than point did. What could be going on here? Could these Paleologs be from regions of the genome that point did not analyze? Or have I made some improper assumptions when simulating and training Frackify? So to do this, I split out all these additional Paleologs Frackify identified, and then plotted the KS values for Paleologs and non-Paleologs. Here we have an example in Brassica rapa, and I was trying to identify Paleologs in this focal WGD. And as you can see, very few of them have KS consistent with the older duplication. I also compared to the outgroup using a similar method, and across the 75,000 additional Paleologs that Frackify identified compared to point, only 489 did not meet this filtering criteria. Similarly, 62% of these additional genes identified by Frackify were not found in the output of point at all. And so punitively appear the majority of these additional Paleologs Frackify identified likely came from regions of the genome that point did not analyze. And that sort of concludes the first part of my talk. And for the second part of my talk, I then wanted to try using Frackify on some actual data sets that I had questions regarding. For this, I wanted to get back to my original questions. Do Paleologs contribute to adaptation in diploids? And to begin exploring this question, I wanted to know whether or not the pattern we saw in Brassica rapa, where candidate genes associated with domestication were significantly enriched with Paleologs, occurred in other species as well, and what was the overall magnitude of this effect and level of enrichment. As a reminder of what a candidate domestication gene list is, I did a literature search looking for population level experiments that tried to explore signatures of domestication in domesticated crops. This includes things like quantitative trait loci, genome-wide association studies, or signatures of selection in the form of reduction of diversity matrices. Similarly, these candidate domestication gene lists had to have gene names which matched known reference and publicly available reference genomes so that I could run Frackify and make Paleolog inferences. Across my literature search, I was able to identify a total of 21 species, representing a total of 5,378 individuals, with candidate gene lists of a total of 29,872 candidate genes, representing a wide variety of domesticated traits, including glucosinolate productions, abiotic stress, biotic stress, flowering times, fruitings, fibers, etc. And between all these species, a total of 3 ancient whole genome duplications. To give you an idea of how I did my tests, I first made inferences of paleologs in each of the genomes and made my null hypothesis be the relative ratio of paleologs to non-paleologs in those genomes. I then compared that to the observed fraction of paleologs in the candidate domestication gene lists and tested whether or not there was a significant deviation from the expected based off the background in the genome using a Fisher's exact test. And here are those results across all the 21 species. Out of the 21, 13 showed significant enrichment in paleologs, and 4 showed significantly less than what we would expect paleologs in the candidate domestication gene lists, with an average enrichment of 9%. And this pattern sort of raised several additional questions in my mind. A, does the level of enrichment vary by the age of the duplication or the level of fractionation in the genome? Do particular classes of paleologs contribute more to this enrichment? What about single retained genes? It's been shown in the past many genes that return to the singleton state are sort of duplication resistant. And similarly, double or triple retained genes are often limited by something called dosage balance. This idea that post-duplication, all copies of a given gene are retained in order to maintain the stoichiometric ratios of the protein being produced. To test this, I used something called phylogenetic generalized least squares, which controls for the phylogenetic relationships between all these species, and we can measure that in a metric called Pagel's lambda. And I did all this in the Caper R library. To put a little context into some of this work, recent work by my lab in 610 species of angiosperms noted that compared the fraction of paleologs left in the genome against the median paleolog divergence of a WD, saw that although older duplications were more fractionated, for younger WD, it was highly variable the level of the fraction of paleologs left in the genome, and this didn't necessarily correlate with the age of the duplication. And so for further testing beyond this point, I did PGLS controlling both for the age of the WG in the form of KS, as well as the level of fractionation in those genomes. The sort of second caveat, I removed 2 species from the analysis that were outliers, sorghum bicolor and Z maize, which is millet and corn, because they had a 40% level of enrichment compared to the average of 9. Similarly, no polyploids were included in this analysis as I was strictly interested in diploids. To begin, I tested whether the fraction of the candidate gene list that was paleologs was predicted by either the fraction of the genome, which was paleologs, or the age of the WGD peak. And I used KS as a proxy. Although you see a slope here, 83% of the signal was primarily predicted by the fraction of the genome that was paleologs, and there was no phylogenetic signal. You can see this on future slides in the form of lambda, a lambda of 0 representing no phylogenetic signal, and a lambda of 1 showing that this trait is evolving under a Brownian motion model of evolution. Next, I looked at enrichment and once again asked whether the fraction of the genome that was paleologs or the age of the WGD was impacting the level of enrichment. And I saw neither a phylogenetic signal nor a significant result. And for my final question, I wanted to know if a particular class of paleologs was contributing more to enrichment. To do this, I used a Mann-Whitney U-test comparing each retention status of paleolog, and single retained genes contributed significantly more to enrichment than the other classes. Interestingly, triple retained genes did not actually contribute significantly at all to enrichment. In summary, paleologs are frequently enriched in candidate domestication gene lists. However, the level of fractionation is more important than the age of the duplication in this process. Similarly, single retained genes contribute the most to enrichment, and double retained genes, although they can contribute, appear to be evolutionarily constrained, which is consistent with the dosage balance hypothesis. For future directions, I want to know why Paleologs are enriched in these domestication gene lists. Our previous work in Brassica rapa noted that Paleologs have increased levels of genetic diversity. However, we don't know if that is true for other species. Similarly, Paleologs may be buffering deleterious variants. And finally, what about natural systems? Are candidate gene lists associated with natural selection also enriched with Paleologs, or is this unique to domesticated species? And finally, what is the overall impact of Paleologs on long-term plant evolution? With that, I would like to thank my lab and the members of it, including my advisor, Dr. Michael Barker, the University of Arizona's Ecology and Evolutionary Biology Department, and then the Botanical Society of America for granting me some funding recently. And with that, I will take any questions. Let me check out the chat. Right, first question.
Thanks so much. That was a great talk.
Thank you very much. What percentage of the candidate gene list came from QTL and GWAS studies versus that used by signatures of selection? So I found that the majority of the studies that I could find primarily were the signatures of selection. However, in terms of the length of the candidate gene lists, the longer lists tended to be those QTL and GWAS studies. I can't give you a direct percentage off the top of my head, but if you want to provide your email, I can calculate that for you, and I'd be happy to send you a response with the precise amount. I'm sorry, I don't remember that specific statistic off the top of my head.
Do you mind if I ask a follow-up question?
Sure.
Do you think like the source of the candidate genes influence whether or not they would be like a paleolog? Like I'm thinking like signatures of selection, like if you're using an FST scan or something where you're looking at divergence, and then you're also looking at like your machine learning program is trained on KS, like could that influence your studies?
Yeah, that's a great question. And that's something we were a little bit concerned about when we were doing the literature search. Originally, we were trying to avoid them, specifically because when you're looking at signatures of selection, oftentimes when you have multiple gene copies, whether it's a paleolog or a paralog, it can sort of lead to a lot of problems, as you pointed out. But in that search, we ended up being limited to only a handful of species. And so we started including them just so that we could have a large enough data set. I'm thinking that might be some of the reasons, like the level of enrichment might be due to some of those issues. But I haven't tested for that as of yet.
Got it. Thanks. Yeah, I don't know if your sample size is like good enough to test like independently depending on like the sets, but just interesting to look into. Thanks.
Yeah, that would be something great to look into. Thank you for pointing it out. All right, I've got a few more questions. Going back to your plots of accuracy versus mean KS, can you comment on the other triangle area of low accuracy? Here, let me share my screen.
In your single retention plot,
Yeah.
there were two areas that looked like they were hotspots for low accuracy, and you
Mhm.
commented on one where the KS was quite high, but there seemed like there was another one.
Yeah, for the single retained genes, there's a little valley. Let me find that slide again so people can see what we're talking about. Here we go.
Yeah, that one. Yeah, so what's going on with that upper one?
With the single retained genes, yeah, right in here. That's what we're referring to. So what my best guess is on that is there's sort of, we had a hard time figuring out why that was, because that's sort of the mystery nature of machine learning, is it's a bit of a black box. We tried to use something called SHAP values to figure out what was going on there. And we didn't get an exact answer, but my current hypothesis as to why that might be the case is in that region for single retained genes, there's like this Goldilocks zone where there's a little bit of overlap between two neighboring peaks that for whatever reason, it's mistaking these single retained genes as double retained genes. I don't exactly know why, unfortunately. That's the main downturn of machine learning, but that's based off the SHAP values. That's sort of what we're guessing is happening. I'm not sure if that answers your question entirely satisfactorily, but
as good as you can. I mean, I trust me, I understand the whole black box with machine learning.
Machine learning, yeah. All right, let me get the chat again. Okay, what was the next one? Okay, I'm going to have to, okay, so Janet left the other meeting. What time is it? So I think we've got.
It's probably time to move on anyway, Michael. The other JW is here, And Jeanette has to miss the second half, and I had to miss the first half. So I look forward to catching the recording of what I'm sure was a wonderful talk. I just caught the last few minutes of it. So thank you very much for your talk. And I guess we will move on. I imagine that Annaliese is here somewhere. And I will get to, I see her. Hello, Annaliese.
Hello. You continuously record both sessions and also over the questions? Just
I'm
checking.
sorry?
Oh, it's still recording. I was wondering if you'd split it into two or it's just the whole thing. Okay.
No, I think the whole thing, I think the whole thing is one recording. Is that not correct? Somebody
That is
know?
correct, yeah.
Okay.
Okay. I'm immortalized asking this question. No worries.
Okay.
All right. I'll share my, should I share my screen or?
Yeah, I get to introduce you.
Oh, okay.
I get to say nice things about you.
Okay.
Well, that's easy to do. Annaliese Mason, I get to introduce Annaliese Mason, Professor Annaliese Mason. She's originally from Australia. She did her undergraduate and her doctoral work in Western Australia. After that, she did a couple of reasonably short postdocs and then moved to, and I'm going to botch the pronunciation, Annaliese, Justus von Liebig University as a research group leader until last year when she became professor and chair of the plant breeding department at the University of Bonn in Germany. So I'm delighted to have her here. Her specialty is in genetics and genomics of hybridization and polyploidy and how that can lead to adaptation. In addition to how we might harness this information to improve crop plants. She's a real leader in this area. And so it's a pleasure for me to introduce her to introduce her talk, which is entitled New Hybrid Brassica Types. Annaliese.
I can't share my screen. Sorry. I need to have the access to.
Uh-oh.
No, you just need to
that?
host.
Who's the host?
It should be good
Michael.
to go now. I changed the settings.
Okay. Yep, that's fine. Good.
Okay. Thank you, Michael.
Okay, all good. Is that fine? Okay, good. So I will talk about new hybrid brassica types, which I previously used to entitle hybrid speciation in brassica until I started running into the speciation literature a bit more closely and realized I don't really want to open up this kettle of fish. So basically, most of our work is about the used triangle of brassica. So you already heard a little bit about this in the previous talk. Well, not really about this, but some of these species. So this is Brassica rapa, which is Chinese cabbage or turnip. Brassica nigra, which is black mustard, and Brassica oleracea, which is the European cabbages, cauliflower, broccoli, kale, etc. And this Korean botanist called Nagaharu Yu established that these have he decided these have the A, B, and C genomes. So these are diploid species, or as we previously heard, Paleopolyploids. And they actually gave rise to allotetraploid species Brassica junctia, which is Indian or leaf mustard or brown mustard with the A and the B genomes, to Brassica carinata, which is Ethiopian mustard and has a B and a C genome, and to Brassica napus, which is oilseed rape, rapeseed, canola, and swede, which has the A and the C genomes and is the most agronomically important of this 6 species. So all of these are actually crops, and all of these have a very nice polyploid relationship, which allows us to do interesting experiments, so to make hybrids and things. So I'm going to talk briefly about 2 experiments from previous PhD students in my lab today. One is about hybridization between the allopolyploids, junctia and carinata. So Brassica junctia and Brassica carinata are interesting because they share the B genome. So this is a set of 8 haploid chromosomes. The A genome is 10 and the C genome is 9. So none of them have the same chromosome number. And if you hybridize these, which is relatively easy, then you can get these BBAC hybrid plants. And so the question was a little bit like, okay, what happens to these plants over time? So we self-pollinated them. This was actually 6 generations worth of experiment. And this is like through self-pollination and selection for the most fertile progeny in each generation. So at the end, we have our S6 lines, 2 different genotypes of the Brassica carinata parent and then 1 junctia parent. So 2 independent lineages effectively. Okay, so our hypotheses were as follows. So I do admit this was kind of a like, let's see what happens type experiment, but potentially it is interesting. So it may be related to how some other polyploids form in the wheat group, for instance. But we start off with this ABBC genome complement. So this diploid B genome and a haploid A and C. And then the question is, with the selection for seed fertility, do we at the end produce new stable karyotypes? So do we have new stable plants? And what actually does happen? So do we get complete loss of both haploid genomes? These are the potential hypotheses. So maybe we just end up with the diploid genome and we lose the A and C. Maybe we retain both haploid genomes, so we get AABBCC somehow. Maybe we get selection for one of the two haploid genomes, so maybe they prefer to keep the A or the C subgenome. And maybe we get some recombination to form a new AC diploid genome at the end. Okay, so this is actually, we could have probably predicted this in advance if we'd thought about it more, but yeah, so regular meiosis occurs in the later generation BBAC hybrids. So in the very first generation, this is basically an indication that these hybrids are very unstable. So they throw a lot of progeny, which have very different chromosome complements. And we did this measurement with meiosis, so this is about 67% bivalent pairing. So this is for the cytogen, if there's any cytogeneticists in the audience, this is the actual cytogenetic configuration. So univalence, bivalence, trivalence, and quadruvalence on average. But then by the time we get to the S6 generation, so 6 generations of self-pollination later, we have highly stable meiosis. So we have uniform bivalent formation. Here's the cytogenetics in case there's any cytogeneticists. So we also found at the same time that the seed fertility in these S6 lines restored itself to the parent species levels in many progeny sets. So we did a kind of selection where we had small progeny sets in each generation, descended through each lineage. Okay, so seed fertility is back to normal, meiosis is back to normal. What's actually happened at this point? Oh, that's not very good resolution. Oh well. Okay, so this is fluorescent in situ hybridization. Sorry about the quality. This is what's happening in the very first generation of meiosis. So this is important because this actually helps to explain also what we found later. So we found so normal BB chromosome pairing. This is what we expect. This is the homologous genome. And we also found an average of 7 AC chromosome pairs per cell in this hybrid type. So this is huge. There's only 9 C chromosomes and 10 A chromosomes. The majority of them, in fact, in some cells, like up to 9 pairs plus a trivalent, were actually pairing with each other. So they're already effectively acting like homologous chromosome pairs in this scenario. So this helps us then explain what happened then to the A and C genomes. We started with 10 A chromosomes and 9 C chromosomes. And at the end, we ended up with these recombined AC chromosomes here. So this is what there's actually 2 copies of each of these chromosomes. Sorry, I shouldn't put that there. But you can see we actually have recombined some of these chromosomes. So we have partial A1, partial C1, A2 is mostly there. Some chromosomes are inherited intact. We have a whole C4 and C7, and then an A8. So these are all homologous chromosome pairs. This is our new stable karyotype. And remember, we had 2 different lineages. This is the other one. So there was absolutely no reproducibility at all. I mean, it was only 2 lineages, but we saw no bias selection for any particular regions or anything. I mean, not that we need 100 lineages before we could see anything probably, but yeah. The other thing we can observe here is that there's more blue than there is red. So there was a bias towards selection of that A genome. I think this is maybe due to the fertility related thing. So from this project, the main conclusions were that selection for fertility, we think, resulted in these new stable hybrid genomes, which were made up of these recombined A and C genome chromosomes. Interestingly, every single copy of an A or C chromosome was present, so every A or C region, every homologous region was present in exactly like 2 copies in the homologs. So there was no loss or gain of any homologous region. So they retained either the A copy or the C copy for every single chromosome region. And this is our result. So the karyotypes produced were also not reproducible, which is potentially interesting, but there was a bias towards the retention of the A rather than the C genome segments. Okay, onto the second experiment. So we have in the used triangle of Brassica, we have the diploid species, so the Rapa, nigra, and oleracea, and then we have the allotetraploid species, Junctia, Napus, and carinata. What doesn't exist naturally is an allohexaploid. So you can make allohexaploids in different ways. You can cross between the diploid and the tetraploid, but we actually had an interesting crossing design which relied on crosses between the allotetraploids. So if you cross between Brassica, Napus, and carinata, then you end up with F1 hybrids, which have a CCAB genome complement. So like our BBAC hybrids in the last experiment. But this time, if we cross them to Brassica junctia, these are actually very sterile, these hybrids, we end up with majority, about 50% of the progeny which are produced in this cross have approximately an AABBCC chromosome complement. So unreduced gametes, so ones which carry the entire CCAB chromosome complement, hybridize with these AB gametes from junctia to get AABBCC. Okay, so then we had the H1 generation and a couple of generations of self-pollination of these hybrid lines. I think we're up to a few more now, but this is a subject for later talks. Okay, so what were these like? So we had very few seeds in the first generations. This is something we observe very frequently when we're making interspecific hybrids. Possibly it's genome shock, but there's like normally a very poor fertility for the H1 generation. After a couple of generations, some individuals though seem to restore fertility, whether this is due to selection because they're originally very heterozygous, or they just get their genome better regulated, we don't really know. So here we can see this over time, these are the different genotypes that we had. Okay, so then we thought, okay, what we'd like to do with these populations is also to potentially map the genetic factors which might be contributing to increased meiotic stability and fertility. So the reason for this is we thought maybe an allohexaploid crop might actually be beneficial, might benefit from extra heterosis or hybrid vigor. But the main problem is if they're so unstable in the first generations, so they might, if they have poor fertility, if the meiosis is not very regular, then this causes problems immediately with this idea. So we had some very small mapping populations, a common problem with interspecific hybrid studies, and then used the 90K SNP array for Brassica, so ABC array. Collected seed fertility data, so number of self-pollinated seeds per plant, a number of seeds per raceme, silique, sorry, and then 10 siliques per raceme. And then for a subset of individuals also did meiotic chromosome behavior to see if this correlated with fertility, which it did, which is nice because seed fertility is much easier to quantitatively measure than meiotic behavior. Okay, so at the end, we reconstructed the karyotypes. I say we, is mostly my PhD student who graduated, Roman. And this is actually what the chromosomes look like. So we have AC translocations in some cases. So here are A2 and C2 or A3 and C3 translocation. These are heterozygous translocations still. We had AB translocations. This was a little interesting. We didn't expect this as frequently because the B genome is much less related than the A and the C genome, so it's more divergent. And we had some just chromosome loss as well. So here a whole loss of chromosome B3 and loss of B7. This little band here is almost certainly not actually present. It will be located on another chromosome. So we don't have bits of chromosomes floating around. They're always attached to centromeres. This is parent plant 2. You can see the same thing. I just wanted to show also this is not highly reproducible. So here's another AC translocation. There's an AB, and then we have a loss of a whole chromosome pair this time. This is a bit worrying because this is unrecoverable aneuploidy. There's not going to get another B4. If it's got one copy of B5, there's a chance some self-pollinated progeny will have 2 copies. But in this case, that B4 chromosome's gone forever. Has to cross with something else. And here again, we've lost A4. It's AC translocations, and this chromosome loss. And here also a chromosome gain. So we can see a dosage compensation effect, which is well known in the Brassica genus, that we have an extra copy of C4. We're pretty sure this is actually 3 copies and not 4, but yeah, it can segregate to 4 if it wants to, and then replacing or compensating for this A4 chromosome. Okay, so we also observe some this bias in translocation direction. So this might be interesting for this audience. So here, this is duplications in each genome. So on the top here in blue, per chromosome down the bottom, so A, B, and C chromosomes, and then deletions per 100 plants in red. And what's interesting here is that we have an overrepresentation of A duplications and C genome deletions. So 2/3 of events were actually A to C rather than C to A. So we're seeing the same selection pressure that we saw last time in the other study, which is a completely independent set of hybrid material. Okay, so our original idea to map for the meiotic stability was a bit challenging. We saw presence or absence of chromosome B3 had an effect. There was some meiosis or some potential QTLs that came up as associated with the fertility, and translocation also here. So in general, as an overview, what was affecting fertility and genome stability in these like extremely de novo hybrid populations? Positively affecting fertility was, interestingly, this translocations of A genome segments onto C genome chromosomes. So this is somewhat of an explanatory mechanism for this bias we keep seeing. 155 more seeds per plant in one of the populations from having 2 copies of the A instead of having an A and a C. So like having extra A homologs. Yeah, we're not really sure what causes this. And then negative was also loss of A7 and duplication of C6. We had a segregating translocation. So this is pretty much the same effect as we're seeing here. And then having more genome rearrangements present has a negative effect on seed fertility. That's predictable, right? Because the more rearranged the genome is, the less likely you are to have good homologous chromosome pairing if you get heterozygous translocations anyway. And then univalent chromosomes, interestingly, had quite a strong effect also. So loss or presence of only 1 copy of a chromosome. So this strong aneuploidy in terms of dosage effect had a negative effect on seeds per plant. So this is in the segregating H3, H4 populations from the parent karyotypes established before. Okay, so at the end, we identified some alleles of meiosis genes which are present in the parents, which might affect the fertility and stability of these allohexaploids. Unfortunately, this really looks like a quantitative trait. It looks like there's multiple loci involved. We're not seeing one major locus. We're actually looking probably at genes or allelic variants or gene copies, or maybe even copy number across the A, B, and C genomes. So we have possibly some interaction here with the allohexaploids, which makes this a little tricky. And it seems probably predict at this point, also from some other data, that the number of functional copies of each meiosis gene may be playing a major role. So validation of this is ongoing and extremely challenging. So I don't know how much progress we will make with this in the short term, but we're probably better off treating this as a quantitative trait rather than as a qualitative trait, at least at the allohexaploid level. Okay, so just as a quick overview, so in Brassica and many other crop species, there's a lot of opportunities to exploit polyploidy and hybridization, as well as crop wild relatives for crop improvement and breeding. So a better understanding of the processes involved in this hybrid and polyploid species formation, interesting enough on its own to this audience, I think, but might also help us to make new crop types, particularly in the Brassica genus. This is the phase labs, picture's a bit older, this is from Gießen, but it has my now Dr. Roman Gaberlein and Elvis Katcher, who will soon be a doctor as well, I believe. So yeah, I would like to thank also these 2 PhD students particularly, and then the lab group in general. So, oh, sorry, that's my, that's the, in case people ask which meiosis genes, which depends on the audience if they're interested or not. But it's in the paper also if you need this information. Okay, I don't know.
Well, thank you very much, Annaliese. Just on time, the fascinating and really whirlwind tour of some of the recent work from your lab is absolutely spectacularly interesting. I have lots of questions, but I'm going to invite others who certainly have questions to put them in the chat or to speak up if you wish. Feel free to reveal yourself.
Also, if I didn't explain something, if I went too quickly, you can also just ask me to better elaborate on something. If something was like really unclear, you're probably not the only person in the audience going like, what was that?
So it might be easier for me to just pipe up than to spend the time typing it out and then repeating it verbally. So super interesting talk. I'm curious about if you are planning on doing any kind of expression work with these hexaploids, because one thing I noticed that you didn't mention there on your list of ones where you saw sort of detrimental effects of, the chromosome rearrangements and things. You didn't mention that nullosomy on, was it B4 that it was missing completely?
B4 and A4, like two different populations, yeah.
So in either of those cases, did those take a serious hit in fertility?
Yeah, so well, I mean, it's hard to say what is a hit in this case because, like, yes, it was, well, I mean, the whole population was missing B4 and A4 because it wasn't present in the parent. So remember, we're talking about the segregating population from this particular karyotype of these three parents. So we can't actually say like they're fertile. I mean, they're making seeds. It's not clear. Probably they'd be better off if they did have that chromosome copy. I think the thing is, they're so redundant, right? So they have like these three genomes. They're already recent. They have relatively recent Paleopolyploidy events as well. They're not.
So that's what I'm thinking. Like
that much
with these higher order polyploids. It seems like these higher order polyploids in a lot of cases, there's so much redundancy that dosage seems to be less important. So I'd be really curious to see, if you find this more commonly, if you're making any other of these artificial hexaploids that are, missing whole chromosomes. Sorry, go ahead.
If we compare it to, for instance, the effect of these kind of events in synthetic Brassica napus, we have a lot of synthetic Brassica napus as well. I didn't talk about this, it's like another project. Then synthetic Brassica napus is much less tolerant than the hexaploids. So then you see a stronger effect, stronger negative effect on fertility of some of these translocations, rearrangements, aneuploidy events.
But the napus is 4x, right?
Yeah, so they're also quite tolerant. We would like to do some expression studies to see what's happening with regards to these translocations and the fertility. So I would like to work out what is happening why does translocation of the A genome onto the C genome give a fertility advantage, and why does this other way give a lack of fertility advantage? I don't know. Is this related to subgenome dominance in some way? Like maybe. But maybe it's also just an effect of actually moving the translocated region to the other genomic environment. I'm not really sure.
Either way, it's really cool work.
It's quite difficult to design an experiment to effectively test this, but we'll try the synthetic napus, which is a little less complicated than that hexaploids.
Well, thank you very much.
Are there morphological differences between the AA, B, C, C compared to the parents? We didn't check for transgressive phenotype segregation, which is probably what this question is really asking. There is much, like that they're larger, so they're bigger plants. You end up normally with like a very large heterosis advantage like we predicted. It doesn't translate into seed yield, it just translates into biomass, and we haven't actually measured this. We're actually planning to measure this next year if we can get enough seeds for a field trial in collaboration with one of the other groups in Bonn who regularly do these kind of biomass experiments. But this was non-trivial for us previously to estimate this for all of these plant species, so for these plants. So I mean, I don't even know like what time we harvest them or like what, I don't know. But yeah, so I think in terms of there was some, there's some there are definitely, there's probably a few novel mutations and a bit of transgressive segregation relative to the parent phenotypes, but we didn't specifically look for this.
Annaliese, if you had to bet, if you had to place a wager at mechanism underlying why, I mean, I don't know if the A genome is a bully or if the C genome is a sissy, or if what's going on with gene expression, nucleolar dominance, network effects. How do you view this?
Okay, it's not nucleolar dominance. So I have a hypothesis that bringing these two subgenomes together, bringing multiple subgenomes together, causes like the cell machinery, which is responsible for regulation of those genomes, to start from the dominant genome to start attacking the recessive genome to some degree. So I'm not sure if this is actually what's happening, but I think there's maybe some issue where we're getting mechanisms which are basically optimized for regulating gene expression, and also or things like transposon suppression and stuff in one genome, or even elimination of chromosomes, like repetitive sequences or something, just like going into overdrive when they come up next to a genome which has a different regulatory balance of this. So I think it's maybe crosstalk between the genomes involved with regards to this regulation. But I don't really know. Like this is just my current favorite hypothesis. It sometimes changes like monthly.
It's funny you use the word overdrive because it's almost like it's a kind of a drive. It's almost like it's a kinematic drive. But for certain translocations.
Yeah, I mean, I think my thinking has shifted recently to thinking that it might be operating also on the translocations as they occur, and that this is maybe something we need to investigate. That we need to look at the mechanism in by way of looking at the subsequent stages of how this would happen. So if we're looking long-term at loss of genes from one genome, we have to look at the mechanisms by which these genes are actually lost, which means we need to think about what's happening to these duplication deletion events, these translocations. Like, are they being eliminated more readily from one genome than the other? Or yeah, this kind of thing. Like, I'm more interested. Yes, exactly. So the C genome is full of transposable elements. So it is 100% like the recessive genome in terms of if you look at the genome structure and you just predict it based on this. So it's much larger, but it has the same gene content. There's more repetitive elements and they're closer to genes, but the gene expression is the same between the A and the C genomes, almost the same. There's very little difference, maybe like a couple of %, and it differs by tissue type. So this is interesting. This destroys a lot of our theories about global gene expression. It's like there's no clear effect in Brassica. You'll actually hit the literature and you can find studies which say that one is expressed over the other, but it's all tissue and genotype specific probably.
Super interesting talk, Annaliese. Thank you very much. I guess I reserve the final question for myself. What part of Bonn is your background from?
Oh, I'm sorry, that's Albany. It's my mother's hometown in Western Australia.
Nice. Yeah, I know. I was trying to be funny.
Yeah. Oh yeah, because it's inland. I'm sorry, I lived like on the coast my whole life and then moved to Germany and now I'm in the middle of nowhere. So I always think like it's totally normal for me to have beaches. I should have a picture of them.
Thank you again, and thank you also to Michael. I look forward to seeing your talk, and we'll see everybody again here, same time, same place in a couple of weeks.